So it's my pleasure to introduce Stefano Panzeri. He is a mathematical neuroscientist and the director of the Department of Excellence for Neural Information Processing here in UKM. His research lies at the interface between theory and experiment and aims to understanding the principles of cortical information processing by developing new quantitative data analysis techniques based on the principles of information theory and by developing computational models of neuronal network functions. And notable works are relevant for the symposiums are quantifying how much sensory information in a neural code is relevant for behavior and cracking the neural code for sensory perception by combining statistics, intervention, and behavior. And it's my pleasure to...
Thank you for the introduction, and thanks to organizer for the hospitality and the invitation, and thanks to all of you for staying so late here to work with us. And before I start the talk, I'll just acknowledge and thank all my collaborators, and our contribution to this work was the computational one. So I'm a computational neuroscientist, so all these students and postdocs that coming from my lab, and also I would like to thank the collaborating labs that provided recorded all the experimental data that I described, in particular Chris Harvey at Harvard Medical School, from whose lab come most of the data, but also Dima Rimberg at NYU. And so the talk, I think what the talk is about, the title is Contribution of Neuron-to-Neuron Interaction to Perceptual Decision Making. So this talk, like the one of Daniele Marinazzo before me, is about functional interaction, functional connectivity. So the correlations of the activity between the activity of different neurons. So this talk builds on the work introduced and pioneered by Daniele that was explained so nice in the previous talk. And the questions are slightly different. In particular, I think what we try to address with our work is how do the functional interaction between the neurons contribute to some of functions that emerge in our population, and we try to individuate those functions in the rest of the talk. But I think we're also interested in how we can infer if functional connectivity is causal or consequential for these functions and for behavior. And here I think I refer to all the caveats that Konrad Kerding put forward in the talk, especially the caveat that's come up if you take the reductionist approach to the extreme and you focus essentially only in one piece on one brain area. And here I would like to show that maybe it's possible to address some of these caveats, and so paint a less gloomy picture for drawing causal or consequential relationship in neuroscience. And also something that I think is specific to this talk is that our work is that it is at the level of neuron-to-neuron interactions, non-interaction between brain areas as measured by functional imaging measures that don't have single neuron solutions. And also is specific to one particular kind of cognitive function, which is that of making accurate decision based on sensory input. So this is called perceptual decision-making neuroscience, and this is the function described here of sampling information from sensory inputs. Here you can think that this information becomes represented in patterns of neural activity, and then based on those pattern neural activity and the information about the sensor that they contain, you have to take an appropriate choice or select an appropriate action for that. And empirically, this is done by recording neural activity with single neuron resolution. For example, in our data, this is done with calcium imaging. So there is fluorescent activity, calcium signals that have peaks or transients when neurons emit action potential. So you can individuate neurons and record action potentials. And usually you do those recordings while in our case, mice are placed with a head fixed on a styrofoam ball, and they can move and navigate in a virtual environment. In this case of this experiment, it was a linear maze. And then, for example, based on a sensory input, in this case was the direction of the sound coming from the left or right hemisphere, take an appropriate action to get a reward. For example, here the appropriate action that the mouse had to select was to turn toward the direction of sound at the end of the maze to collect the reward. So the problem we study is how the interaction between neurons in a given brain area somehow help or maybe don't help this function, which is to represent sensory information and then based on the sensory information select an appropriate action to get a reward. And in most of this data that I show, those were recorded with these calcium imaging techniques from posterior parietal cortex of mice. And posterior parietal cortex is, I think, is a good place to look at the relevance of sensory signal, because it's what's described an association area or an interface area, so an area that somehow is known from various studies to convert sensory representations into goal-directed actions. So I think it's a good place to look. And so correlation between the activity of neuron, functional connectivity, Daniel already introduced it so beautifully in his talk, but it's the activity, the correlation between the activity that you measure if you record simultaneously different neurons in the brain. If you do that, if you record simultaneously many neurons in the brain, essentially you find that almost all pairs of neurons are correlated. So they depend, the activity of one neuron depends on the activity of other neurons. And this is because the neurons are connected. This is by synapses. This is because they covary for various reasons, like changes in the modulation and so on. So there are various reasons why they covary, but essentially all of those neurons in the brain covary. I mean, this has been reported for many years with robust findings, and it's found at many scales, from the single neurons to the correlations across areas that were described before. And I think there is a kind of a lot of debate about what this function is. And essentially how you can quantify and see correlations in the data. This is something we will use later. So say that you have here just two neurons, neuron 1 and neuron 2. So you can put dots on the plane. Each dot would be the activity of those neurons in a trial. For example, this one would be an activity of the neurons, and you can color them by condition. For example, in the task that I explained, you have to decide whether to go left or right, or you see stimulus coming from the left or from the right. So you can color the dot of neural activity in a given trial, so in a given execution of the task, by the condition in which they were recorded. And each dot is a trial, and this is the cloud, so it's a probabilistic process. There is a lot of variability, but if you plot them one against the other in the scatter plot, usually you find this kind of bananas or ellipses, so elongated distribution, which means that you can predict the activity of one neuron given the activity of the neuron. They are correlated. You can maybe do things shafting, so this will be the shape of the distribution if neurons were not correlated. But because they're correlated, you have those kind of elongated distribution, which you can predict activity of one neuron from the activity of the other. And so what we are interested in is, what's this function of those correlations? Why are they there? And do they have a consequence downstream on behavior, on brain functions? And the traditional way to try to look at this problem is to look at how this correlation shape what we call sensory encoding, so the encoding of sensory information. Sensory encoding for us is the transformation between the external stimuli, the sensory stimuli that you use to make your decision, and patterns of neural activity. And the idea is that the more information you have in neural activity about those features of the external world, the easier for you will be to take the right decision. That's why people look at that. And there are analytical ways, mathematical ways to study this. I think in a simple way, what we do is to build classifiers, decoders of multidimensional patterns of neural activity that can tell us which stimulus condition it was. And those essentially are kind of linear hyperplanes in the multidimensional activity space that tells you if it is this side of the hyperplane, it's the blue stimulus, the blue condition. If they are on the other side, it's the red condition. And then you can study the role of correlation by various techniques. And the simplest one is using essentially various tricks to destroy correlation in the data, leaving the single neuron properties the same. And then you can compare whether destroying the correlations, the functional connectivity, and leaving the single neuron properties the same, increases or decreases the information they carry about the stimuli that you have to discriminate in order to make an appropriate choice. And essentially, if you study mathematically how the correlation between the neurons affect information, you find that in most, not all, but most cases, you get a little disappointing result. You get that having this functional interaction between the neurons reduces the amount of information available in neural activity. And this is explained in this cartoon. And so here is like the scatter plot that I showed before. So the joint distribution of activities of two neurons simultaneously recorded. And each ellipse is the distribution of the activity of neurons to each of the two conditions. For example, this would be stimulus left, and this would be stimulus right. And if there is no overlap between those two distributions, then it means that you can always, whenever you have a population response of those two neurons, discriminate perfectly the condition, and so make always an accurate choice and get your reward. If there is instead a lot of overlap, if you have something like this, a neural response in this region, you would not be able to tell which condition it was based on neural activity, and so you would fail to, you would have to make a random choice and you would fail to get a reward. And in most cases, because correlation between neurons are positive, so neurons, when one neuron fires more, the other neuron fires more, and because these neurons that have positive correlation in most cases have also similar tuning to the stimuli, then having correlations gives more overlap between the distribution than not having them. So as a result, when you shuffle the data, so when you destroy correlations, you get that you have more information in neural activity. So this would be the decoding of the stimulus based on neural activity that you get. I don't know, this is about 50 neurons that we recorded here in posterior parietal cortex, and you get from those 50 neurons a decoding of about 65% correct, and sorry, 60% correct with the real data. If you leave the single neuron property the same, but you destroy the correlations, you get more information. So having those correlations between the neurons disappointingly reduces information. So it seems like those guys are really bad for behavior. And this is an example from my lab with these 50 neurons and so on, but people have done also recordings with more neurons. For example, this is recording from primary visual cortex, discrimination of different stimuli with different orientations. And if you remove correlations, you get a lot of information as the population size grows, but with real correlated responses, information doesn't grow. And so it limits really the amount of information you have available to make decisions in a local area. And because of this, people kind of concluded, studying this, that considering only encoding information in a given area would lead to suggest that informations are bad and they limit behavioral performance. But as Conrad has, I think, said in his talk today, I think quite nicely, if you only consider a local area without putting it in this context, you are in a danger of maybe missing out the real function of the population that you address. And I think in this case, if you only consider encoding, you are missing out if correlations are really consequential to forming behavioral choices. If the readout of the information in a given area from the rest of the brain is totally optimal, then the more information you have, the more information would be used by the rest of the brain, and the better will be the decision you use based on that. But for example, if the readout is not optimal, or if maybe it is, then this will not be the case. So having a lot of information would not help you necessarily. And also it's possible that the activity you are considering has really no consequence of the behavior you are considering. From observations, it's very difficult to address that. And one way that made us think that maybe we have to address this issue was that we did a very simple kind of control analysis. And we thought that if these correlations are really bad for you, for behavior, then you can, you have a simple prediction that since they are bad, when you have more correlations, you would have a lower probability of making an accurate choice. Whereas if you have less correlations, you would have a higher probability of making an accurate choice. But then if you look at all trials, you divide trials or actions in which the mouse made the correct choice. And from trials where the mouse made an error, you get exactly the opposite of this prediction, that you get higher correlation when the mouse makes correct choices than when the mouse makes incorrect choices. So this is kind of paradoxical, and these are odds with this view that you get if you look at local encoding only from the given area. So you have, at the same time, stronger information during correct behavior, suggesting that they're good for you, they help you to make choices, but you also have less information because you have correlation. So how can you solve this paradox? And in our view, I mean, this is similar to what Conrad kind of suggested. A way to try to address this paradox is not only at the encoding of information in this local area, but also to try to kind of make an idea to map what is the downstream effect of this activity. So considering essentially this activity not only locally, but also in the context of the rest of the brain. And there are two ways to do it. And the one that you can do if you don't have access to the activity of other areas is to look at what would be the kind of overall global readout of this information. And one way to do it is essentially to try to map you cannot maybe map the flow of this information really from one synaptic step to the next, but measuring behavior and the relationship between information neural activity and behavior, you can kind of make a good model, make a good understanding of what is the overall readout of this information by the rest of the brain. That's what we try to do. And to explain those paradoxical effects of lower information with correlations in a local area, but better behavior, so higher correlation with when the mouse makes correct decisions. We decided to investigate this operation, which is the readout, so the statistical map between the sensory information and neural activity in a given area and the behavioral choice. And the traditional hypothesis, that is like the reductionist, essentially is similar to the reductionist hypothesis of considering activity in a region without really caring about how it is read out. That kind of is equivalent to assume that there is an optimal readout, so all information encoded in a given area is utilized for behavior on a single trial basis. So if that happened, it would make the choice on the most likely stimulus computed from the posterior probability of stimuli given neural responses. And if that was the case with neural responses like we have, where correlations decrease the information because they increase the overlap between the two distribution of the stimulus of the condition that you have to discriminate. So if you have data, then you would have less information with correlations, and because there is a proportionality between information and behavior, and then the readout map would only depend on the total posterior probability in each trial, so we uniform in probabilities in the neural response space. And then in that case, you would have that the behavioral performance, so the accuracy in discriminating, so behavior would be higher when you remove correlations than when you have it, because it's proportional to the encoding. But it will also predict that, unlike in our data, you have stronger correlations in error trials than in conditions in trials where the animal made incorrect decisions. So we can rule out these based on our data. And instead, I think the hypothesis we made is that the downstream readout of this information works better if to population, to neurons, whatever, provide consistent information about the stimulus. Consistent information about the stimulus about the neurons is that mean that both neurons, by their activity, indicate the presence of the same stimulus. And what correlations do is that they may decrease the information, but they increase the probability that those two neurons, encode consistently the same message. And in this kind of map in two-dimensional space, this would be those two regions here, the top and bottom corner, would be the regions in which neurons encode information consistently. So, for example, in this region, Both neurons have very low firing rates and indicate the presence of the blue stimulus. And in this region, both neurons indicate the presence of the yellow stimulus. Whereas in this region, one neuron would have a low rate and the other neuron a high rate. So one neuron would indicate blue stimulus, which is the low activity, and the other neuron would indicate the yellow stimulus, so inconsistent. And if you compare these with correlation with these, you see that here you have maybe more overlap between the distribution, so less information, but a higher proportion of trials that have consistent information. So we made the hypothesis that if you are in this region, if the responses are in this region, they are better read out. So there is a higher probability that the choice of the animal follows the indications set by neuron population activity. Whereas if the responses are not consistent, so each neuron tells a different story, then you would have a lower probability that the brain follows the indication of neuron population activity. And if you take this model, then you would predict that you have higher behavioral performance with correlated data, so correlations help behavior, and also you would have stronger correlations during correct behavior than in our data as in our data. And so we made this assumption, and then we kind of made a model that allowed to demonstrate this very quantitatively on a trial-by-trial basis. So we made a statistical model of the choice of the animal in each trial based on the information encoded in neural activity. This term here would be the, total information carried by neural activity in a given trial that allows us to reconstruct the stimulus. And we express in this model the animal choice as a function not only of the total information in neural activity in the population, but also with an interaction term and a linear term that, to cut a long story short, quantified how much the total information, the readout, so the effect of total information on choice was modulated by the correlation or the consistency between the neurons. And in our statistical model, we found that adding this term increased our ability to explain choice of the animal on a trial-by-trial basis, so it has an explanatory value. And also that essentially the shape of this term would be that it would amplify the readout of information when the neurons gave consistent information, and would suppress the readout of information when the neurons given inconsistent information. So for every bit of information in a population code in the brain, you have a higher readout of that information, a higher utilization for behavior of that information, if all the neurons express the same information. And this is a suboptimal computation. One way of thinking like that is, one example that I make is say that you have to gain information about physics, interviewing neuron 1, which is Einstein, and neuron 2, which is me. If you are optimal, you would not listen to me and you will listen to Einstein. But if you are suboptimal, you would tend to follow the advice if the two of us give the same advice. And you would maybe be confused or not necessarily follow the advice if it is inconsistent from the group of people you are interviewing. I mean, it's a suboptimal computation, but it kind of makes sense. And I think one interesting feature about this is that you can then use this model, this readout model, which is a faithful model of how patterns of neural activity inform choices, to predict what would happen if you are able to destroy this correlation with the neuron, which is difficult to do experimentally. And we did some mathematics to do that, and the idea that you put together the information that you have about the encoding, so local encoding, and this readout, and you can predict the overall effect on behavior. And the prediction here is that having the advantages of interaction between neurons to facilitate readout overcome comfortably the disadvantage that you have because they reduce information. So it is convenient to have some level of of correlation, because maybe you lose some information, but you gain in the way that the information is utilized. And I think this is also an example of how it is possible, even with the limitations of neuroscience, to put the activity of neurons in one area in the context of activity of neurons in the other area, so go beyond this reductionist approach, and I mean, gain better information, address some of the caveats that Conrad talked about. And also, as I said, other caveat that were put together is that when you observe data, since you don't have all the neurons in the brain, it's difficult to establish causation. And one way to do it is instead to intervene on the variables. And this is possible because my collaborators are doing optogenetic experiments with the photon microscope, where you can, with front wave engineering, put optogenetics. So those are opsins that allow you to activate neurons when you shine blue light. And you can do it really with the resolution of one neuron, so you can stimulate arbitrarily patterns of neuron depending, for example, of their preference, whether left or right, and so on. And with doing these experiments, you can really probe, for example, the theories that we said. If you, for example, put a couple of bits in the brain by stimulating, for example, 0.2 bits, 0.5 bits in the brain by stimulating one neuron with a lot of information, and the opposite neuron that gives the opposite message, or if you, this will give you, say, 0.5 bits of information. But if you do it stimulating two neurons with small information, but in a consistent way, for any bit of information you inject into the brain, you would have a higher readout of the information, so you'll be able to influence the animal more with this pattern than with that pattern. So this kind of formalism also creates specific prediction that are beginning to be testable. And we couldn't do it yet here by influencing neuron this way in a discrimination task, but we could do it in a paper that we published in Science a couple of years ago in a kind of simpler experiment where there was no sensory stimulus, so the detection of neural activity that is imposed on the brain autogenetically is a little simpler experimentally, because when you do those kind of experiments and you have the sensory stimulus, the information you superimpose in the brain is much smaller than the one that is generated by the senses, so it's a bit difficult. So something that is simple to do now is to, for example, reproduce, this was an experiment done in the olfactory bulb, and initially the mouse, this was done by the lab of Dima Rimberg, and we did the mathematical analysis. The mouse has to recognize the presence of an odor. Then the odor is taken away and is replaced with a pattern of activity generated here by essentially a spatio-temporal sequence of activity in the brain that is similar to the one evoked by odors. And then you ask the mouse to discriminate between this orderly pattern that simulate the presence of an odor from a non-random perturbation, and a non-random stimulation with the same duration, just different either locations or times or different spatio-temporal structure. And the mouse learns to do that, and then you can introduce like a small percentage of perturbation trial where you slightly perturb the target, and you more and more perturb the target until essentially you test the boundaries by which this target becomes this one in the perception of the animal. It's a way to map essentially perceptual discriminations, distances in patterns of neural activity in a causal way. And to cut a very long story short, by manipulating all these patterns, special temporal patterns of neurons in a way, and building a model of the behavioral reports of the animal based on the patterns of neural activity, we asked the same question that we asked observationally. So if correlations between the patterns of neurons enhance the ability of the animal to correctly detect the pattern, and the answer was yes. So there were kind of interaction terms different from linear that increase the probability that the animal will be able to extract information from the brain. And I don't know if this is a causal proof that this interaction between the neurons enhance the behavioral readout of information. In the paper, we call it consequential, because I think we were focused on the downstream effect. I mean, there is a relationship between cause and consequence. And I'm not a philosopher, but also this is something that I would like to discuss, whether those experiments reveal consequentiality or causality. And also something that we are trying to do to relax or address this caveat of not looking at brain areas in isolation in extremely reductionist approach is that what we usually do, as I said, is we look at the information encoded here, regardless of the rest of the brain. But we don't know whether this information is utilized or read downstream. And one other way to kind of address this is to use retrograde stressors. So those are viruses that have a color that are injected in anatomical targets of the region that you record from. And those viruses happily travel back to the region. And then you see them in your field of view with the color that you put in the viral vector. So here, for example, in the field of view, green would be this calcium indicator of the activity of neurons. And you have a halo of blue in those neurons that project to the same target. Those neurons are not organized like spatially together. And if you do that, then you can individuate population of neurons that transmit to the same downstream target. And you can argue that this is really what is read out in the brain. It's not what is encoded locally, which might or not be utilized, but is what is transmitted to downstream targets. And then you could do same analysis as before, but we can do it separately for the nearby neurons, which would be those neurons that are not labeled, so they don't necessarily project to the same target, and the neuron that instead project to the same anatomical target. And there are two things that we found. If we look at like neurons in the same neighborhood, but either without that don't project to the same downstream target, or they do. And the one difference is that you have stronger functional interaction between the neurons that project to the same target, which is compatible with this proposal we had that the correlation enhance the downstream information processes, and also that also the gap between correct and error trials and information strength is larger for the same projected neurons, which suggests that this is again consequential for making correct decisions. And the other thing is that we didn't look only at the strength of interaction between those neurons, but also how these pairwise interactions are organized at the network level. And this is a little different from what Daniele explained in his talk. Daniele concentrated on higher order interaction, which is what you cannot explain by combining linearly the pairwise interaction between the neurons. Here we studied how the pairwise interactions, so the links between the neurons, are organized at the network level. And I'm not an expert in graph theory. I mean, people like Klaus here are, know much more than me, but something that I think I learned and kind of makes sense is that if you want to look at network structure of pairwise interaction, the simple quantities informative that you can look at is the distribution of triplets. So you may have that, for example, you may have groups of neurons with positive interactions clustered together, and then those are connected, say, by negative interactions, or maybe you have a random structure or the other way around. And by looking at which type of triplets prevail, you have an idea of what kind of organization you have. And if we looked at distribution of triplets in neurons that don't project to the same target, and compare them with the random network where you essentially have the same number of links, same sign, but you just shuffle them, then you have that there is no difference between the randomized network, the null model, and the actual distribution of triplets. Whereas if you look at, so there is no structure in neurons that don't project to the same target. But instead, if you look at neurons that project to the same target, then there is an excess of triplets that whose sign of interaction makes plus, so either plus 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 or plus minus minus. So it's a configuration like this essentially. But that only happens, in neurons that project to the same target, and only when the animal makes correct decisions. No structure, even in neurons that project to the same target, when the animal doesn't make a correct decision. So this is again a suggestion that this structure is consequential for behavior. Then we ask, why this might be the case, because that's a little bit abstract. And so something that we try to do is to try, in the bit of the spirit of this paper Olaf Sporn that was cited by Daniele in the previous presentation, is to try to relate the amount of information about those stimuli that have to discriminate, for example, the direction of the sound in neural activity, relate that thing, which is what you use to generate behavior, to these structures. And to cut a long story short, we did a little bit of mathematics to try to express the information as a function of the single neuron properties and terms that relate to the interaction between the neurons. And essentially, we were able to do some mathematics to isolate the contribution of structure from the contribution of interaction, but without considering the structure. So just, and to cut a long story short, I mean, doing something which is for the people of you that come from physics is really very much similar to, you know, Feynman diagrams. What we found is that triplets contribute to the structure information and contribute with this sign rule. So they give a positive contribution to information if the product of their signs is plus, and they contribute negative information if the product of their signs is minus. And please remember that those were the neurons that are enhanced during correct behavior and neurons that project to the same target. So the good guys are enhanced, they carry more information, and the bad guys that decrease information are reduced. So if you do then the calculation of information carried by neural activity, and you see that if you consider neurons that project to the same target during correct behavior, then you have this positive contribution of information which scales supralinear with the population size, as Daniele explained. And that kind of accounts, I think, suggest that correlations, again, this interaction with the neuron are not that bad, but you see them only if you somehow track how those functional interactions relate to the transmission of information, either with respect to the total behavioral output, or in this case, the downstream effect to the next projection target. And also for me, it's an indication that microscopic interaction structure is organized not to encode information locally, which is something that maybe is nice for us, but is not really used, but is organized to boost the routing of information to downstream targets, which I think is an operation that makes sense. And I don't know how much time I have, but fortunately for you, I think this is the last slide. And those things that I described as functional interaction are not necessarily in one-to-one correspondence with synapses. So we are trying actually to address this question and map it. And the way we do it is that for one of those mice that made the experiment, it was possible to do electron microscopy of the same neuron that we image, and we could reconstruct the connectivity of the neurons and relate them to the functional properties, the preference with respect to the stimulus and so on. And to cut a long story short, it's possible to kind of find interesting structure between those neurons, in this case, a motive of opponent inhibition that seems able somehow to support those calculations of enhancing information that I described. And I don't know. So I think maybe the take-home message of this is that I agree with the conclusions of Conrad Cording that there are dangers in taking an extreme reductionist approach and looking at the piece of information in the brain without putting it into context. But I think I would disagree that it's not possible really to consider any of the, to address, I mean, the caveats that were raised. I mean, it is possible to address them, and it's important to address them. And if you do that, you can get a reasonably clear idea of how information encoded in a piece of the brain is utilized in the kind of wider context of the rest of the brain, and understand how it contributes to behavior. And the conclusion here also seems to be that the design for this interaction is not only based on optimizing information representation locally, but also to make sure that this stuff is transmitted safely downstream to generate behavioral outputs. And Thank you very much for your attention.